Classification of brain MRI tumor images. Abstract medical image processing is a challenging and plays many important roles in several applications. Computer-aided detection, CAD, provides a great distribution in detecting and diagnosing several diseases precisely, such brain tumor which is our field of study in this paper. By using machine learning algorithms, we can demonstrate automated tumor detection, as implemented in this paper. With this method, diagnosing may perform with high quality, and less effort and time. The purpose of this paper is designing a system that will automatically classify the brain images in terms of normality and abnormality. This system incorporates with several stages such as pre-processing, feature extraction, feature selection, classification, and performance assessment. MATLAB program was used for designing and implementation. In the end, the designed system successfully classified the two classes, normal and abnormal, with accuracy of 100%. Keywords: brain tumor, MRI images, normal, abnormal, machine learning-based techniques. Introduction. Growth of cancerous cells in the tissues of the brain is a brain tumor 1. It can be benign, non-cancerous, or malignant, cancerous. The benign tumor may not spread to far areas or grow into nearby tissues, but they can destroy, press, grow on normal tissues, which may cause harmful or even life-threatening damage. Malignant tumors can spread throughout the body and damage other structures too. So benign or malignant, all brain tumors are serious because they will eventually harm and damage other structures within the brain. 3. According to the WHO classification of tumors, brain tumors are classified into class I, II, III, or IV based on disorders of the cells they contain. 4. Lower class tumors such as class I and II tend to develop slower and less likely to grow into adjacent tissues. Higher class tumors such as class III and IV tend to develop quicker and grow into nearby tissues. 2. Brain tumors are categorized into primary tumors, start in brain tissue, and secondary tumors, from another area of the body, reaching the brain. 3. About 30% of brain tumors. Meningioma and glioma are most known types of main brain tumor. Almost 30% of brain tumors are meningioma. It can grow and press against the brain. Most of meningioma tumor are benign and most can be removed with surgery or treated by radiation. 5. Glioma is a group of tumors start in the brain's glial tissue. Glioma group contains astrocytomas, brainstem gliomas, ependymomas, oligodendrogliomas, and optic gliomas too. Sophisticated imaging techniques can pinpoint brain lesion. Magnetic resonance imaging, MRI, and computed tomography, CT or CAT scan, are diagnostic tools. Magnetic resonance spectroscopy, MRS, is used for examining the nature of the tumor observed on the MRI and to analyze the tumor's chemical profile. To recognize recurring brain lesion, positron emission tomography, PET, is used. Sometimes, the neurosurgeon uses the biopsy to create a certain diagnosis of a brain tumor, and the pathologist decides the final determination whether any benign or malignant tumor is appearing and grade it accordingly. 6. In 2020, around 23,890 malignant tumors, 10,300 females and 13,590 males, will be analyzed. If benign, non-cancer, tumors were counted, these numbers would be much higher. Caused by brain and spinal cord tumors, around 18,020 individuals, 7,830 females and 10,190 males, will die. Generally, less than 1% is the possibility that an individual will have a malignant tumor of the spinal cord or brain during a lifetime. The hazard of developing any sort of spinal cord or brain tumor is a bit higher in females than males, but it's slightly higher for men than women when it comes to malignant tumor. This is explained by that some sorts of tumor are more present in one gender than the other. 7. To recognize forms and features indicatives of tumors and other disorders, computerized algorithms are used. In concept, there is a similarity between biomedical image processing and signal processing. They are similar in terms of enhancement, analysis, and displaying images taken by nuclear medicine, ultrasound, X-ray, MRI, and optical imaging technologies. To automatically analyze and distinguish what may not be observed by human eye, image processing software is used and the basic eyeballing and X-ray is abolished. Aspects as volume, diameter, and vasculature of a lesion or organ are being determined via image analysis and processing associated with the suitable image technique and diagnosis. 8. Literature review. Aranitha 1 and D. Siva 9 proposed a random forest classifier system-based brain tumor detection and segmentation method. For eliminating and detecting noises in brain image, adaptive medial filter is used. Via texture features, features are extracted from the denoised brain image, such inverse difference moment, IDM, variance inertia, angular second moment, ASM, energy, dissimilarity, and homogeneity that are acquired from gray level CO occurrence matrix, GLCM. Classifier such. Random forest, RF, is used to segment the original brain image into normal or abnormal. For severity diagnosis, RF classifier used weighted voting method to segment the brain images. Figure 1 shows the final result of the isolated tumor as pixel segmented image, A, and the spot of the detected tumor of the input image, B. Pushpa Br. F. Louise 10 suggested a methodology where a framework is built to classify and detect the tumor. The proposed method passed through stages: pre-processing, segmentation, masking, feature extraction, and classification. Pre-processing images done through median filter and Gaussian high-pass filter. Morphological operation was used to separate the tumor and the normal region. Feature that are extracted are symmetrical, texture and grayscale using multiple extraction methods like DWT, discrete wavelet transform, techniques. Support vector machine, SVM, classified the tumor as benign, malignant, or normal with accuracy of 99%. John Peter, J., and Panachami, in 2019-11, developed an image fusion and CO-active adaptive neurofuzzy inference system, CANFIS, as a classification method for brain tumor detection and segmentation by combining with the principal components analysis, PCA, and discrete wavelet transform, DWT. Their work contains 4 steps. Step 1. Brain MRIs database acquisition used a dataset of 66 brain MRIs into 4 classes, e.g. normal, glioblastoma, sarcoma and metastatic bronchogenic carcinoma tumors. All the brain MRIs was in axial plane, T2 weighted and 256A 256 pixel. Step 2. Image segmentation using fuzzy C-means, FCM, to separate the different normal brain tissues. In step 3, feature extraction using discrete wavelet transform, DWT, and using principal component analysis, PCA, method to estimate the original extracted features with degraded dimensional feature vectors. Last step is classification using deep neural network, DNN, classifier. Suhag, S., and Saini, L12 improved a system that automatically detect and classify brain tumor using MATLAB GUI tool into normal or abnormal, then classify the abnormal into gliomas, metastasis, and astrocytoma. The proposed system started with pre-processing the image to denoise and smoothing the edges using median filtering. Then applied fuzzy C-means, FCM, for segmentation method. A gray level CO occurrence matrix, GLCM, technique used to extract feature and the useful feature was selected by sequential forward selection, SFS. SVM classifier has been used to classify the images to tumor or non-tumor, and multi-SVM is used to determine the type of tumor, whether it is gliomas, metastasis, or astrocytoma. Jayakandran and Ardana Skaran 13 suggested a hybrid algorithm for recognizing brain tumor in MR images. The proposed algorithm consists of pre-processing, segmentation, feature extraction, feature reduction and classification. In pre-processing stage, the background noise, low frequency removed. The segmentation of brain tissues is done by skull stripping and features extracted using the gray level co-occurrence matrix, GLCM. The fuzzy-based support vector machine classifier, FSVM, classified the brain images into tumor or non-tumor with 94% score in accuracy. Mins et al., in 2017-14, proposed a scheme for classification of brain tumor types from MR images. Threshold-based image segmentation was performed along with processing like RGB to gray color space conversion and filtering for noise removal. Then GLCM-based textural features were extracted from the segmented images. An adaptive boosting technique called AdaBoost outperformed neural network with 89.90% accuracy. Kainani et al., in 2020-15, optimized a computer-aided detection (CADE) system to develop accuracy of tumor detection and to reduce radiologists' workload by giving more quantitative, objective, decisions added to the qualitative, subjective, evaluation of radiologists. Databases were prepared by 3 different ROIS sizes. Their CADE calculated parametric features extracted from ROIS using statistics, histogram, GLCM, and wavelet techniques. To study the significance of features, sequential forward selection, SFS, technique is used. Many types of k-nearest neighbor, KNN, and support vector machine, SVM, classifiers were trained to distinguish between normal and abnormal ROIS. Umar et al., in 2019-16, discussed how to observe the abnormal structures of human brain image. Their proposed mechanism consists of various phases: pre-processing, segmentation, feature extraction, and classification. They used filtering algorithm for pre-processing, clustering algorithm for segmentation, and gray level co-occurrence matrix, GLCM, for feature extraction. By using ensemble classification, automatic brain tumor step is performed. This stage distinguishes brain images into tumor and non-tumors. Goetker et al., in 2016-17, proposed a brain MRI image classification system. In proposing work, image pre-processing is done with median filtering and skull stripping method to remove noise and tissues from medical images. It shows better performance. The features were extracted using GLCM techniques and a linear SVM is used as a classifier. Results of sensitivity 91.52%, specificity of 67.74% and accuracy of 83.33%, was given by SVM approach. Megha et al., in 2019-18, presented machine learning-based approach. In pre-processing step, they performed skull stripping to remove all extra tissues in the skull images. For feature extraction, gray level concurrence matrix, GLCM, was used and then selecting the prominent features. For segmentation of brain images and recognition of tumor using support vector machine, SVM, classification method. Reason of SVM is to separate the nonlinear transformation into a linear transformation using kernel functions of SVM. They used Gaussian kernel that has made the classification very smooth and convenient. They argued that tumor detection was faster and more accurate comparing with manual segmentation carried out by radiologists. Results are 83.3% accuracy recognizing if the tissue is normal or abnormal. Figure 2 illustrates the original image, A, and the final output image after performing skull stripping and threshold segmentation. Methodology. In machine learning, there are some sequential steps must be followed in order for the CAD system to perform properly. Figure 3 shows the processing blocks contained in our CAD system. Database source. To conduct this study, data samples were accumulated from Kaggle website 19. These samples contain 144 MRI images of both malignant and benign cases. The samples were of different resolutions. These datasets were categorized into 72 normal, 72 abnormal, in which 98 ROIS were used for learning and 46 ROIS for testing. Pre-processing. We manually cropped a region of interest, Rio, with size of 3 to 832 pixels to concentrate our work mainly on the damaged area for further processing. This size was a result of many experiments where original image sizes were implemented but algorithm performance was best observed with lower size samples. ROIs were imposed for two different transformation techniques, which are radon transform, RT, and discrete cosine transform, DCT. Features extraction. Figure 3, CAD processing blocks. We have conducted many features extraction functions which act as a mathematical description of samples characteristics to help classifiers to distinguish normal from abnormal cases. After applying cosine transformation to RIOs, we have investigated 23 features and only 17 passed the t-test with p-value less than 0.05. Statistical features on ROIs. Those features are mean, STD, square of STD, mode, median, percentile Schwantile, third moment, skewness, kurtosis, max, min. GLCM features. Level of 32 and shift or distance, D equals 1, in all direction, angles equals 0 a degree, 45 a degree, 90 a degree, 135 a degree, 180 a degree, 225 a degree, and 270 a degree. Those features are contrast, energy, correlation, homogeneity. We excluded features that didn't pass the t-test for the p-value, must be 0.05. For evaluating the classification, all classifiers were assessed based on performance metrics such as accuracy, sensitivity, and specificity. Classification. After implementing features extractions and selecting the best performed ones, we began the classification, which contains two phases. In the learning phase, the data clearly represents the nature of ROIs and teach the classifier whether it's normal or abnormal. During the test phase, the classifier works based on the previous taught instruction. We have used two types of classifiers as shown in table below. In this step, the data is classified into two classes of brain images, normal and abnormal images. In this paper, 9 classifiers were used and compared. The best accurate classifier will be selected for any further processes on this approach. The classifiers are SVM classifiers that has discriminate function of the type RBF, polynomial, Gaussian and linear. The other classifiers that are used are KNN classifiers that have K values of 1, 2, 3, 4 and 5. Results and discussion. Results were obtained after implementing 3 trials with 2 different transformed ROIs. Features were first extracted from original ROI, Oreo, then from radon transformed ROIs, RT, and finally from discrete cosine transformed ROIs, DCT. The testing ratio for the database was almost 32% out of 144 ROIs, with 23 normal and 23 abnormal. MATLAB software was used in this approach. Each trial was assessed with 5 performance assessment metrics: sensitivity, specificity, positive predicted value, PPV, negative predicted value, NPV, and accuracy. The following graph, figure 4, indicates that the SVM did not perform well with neither original untransformed ROIs nor with the radon transformed ROIs. Accuracy comparison of original ROI, random, and discrete cosine transforms. Discrete cosine transform. Discrete cosine transform gives the best results for detecting brain tumor in 4 types of support vector machine classifier: SVM RBF, SVM polynomial, SVM Gaussian, and SVM linear, and 5 types of K nearest neighbors classifier: KNN1, KNN2, KNN3, KNN4, KNN5. Appling t-test obtained 17 useful features, p-value 0.05, showing in figure 5. Figure 6 shows that the best classifier was SVM polynomial with sensitivity, specificity, PPV, NPV, and accuracy equals 100%. Followed by SVM RBF, SVM Gaussian, KNN3, KNN4, and KNN5 with accuracy equals 98%. This result was obtained using 17 features distributed as first-order statistical, second-order statistical, and higher-order statistical. These results indicated that the DCT is useful with multi-order feature extraction. Table 2 illustrates the previous studies related to tumor segmentation techniques and gives a brief comparison between them. Our proposed method shows outstanding results in terms of accuracy, sensitivity, and specificity. Conclusion: this study develops a CAD system to get better detection for brain tumors. 144 MRI images database, 72 normal and 72 abnormal, were resized into 3:32 ROI. 3 experiments with 2 different transformed ROIs were applied for results comparison. 17 features were extracted from each experiment and then were used for classification. Features were of first-order statistical, second-order statistical, and higher-order statistical features. 2 main classifiers were used for classification. Support Vector Machine Classifier, SVM-RBF, SVM Polynomial, SVM Gaussian, and SVM Linear, and K-Nearest Neighbors Classifier, KNN1, KNN2, KNN3, KNN4, KNN5, are the ones applied in our paper. Each classifier was assessed with 5 assessment parameters: sensitivity, specificity, PPV, NPV, and accuracy. Best results were observed from the discrete cosine transformed, DCT, ROIs associated with the SVM polynomial classifier of 100% score in sensitivity, specificity, PPV, NPV, and accuracy.